Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin our question and answers, and I'll ask your questions to our speaker. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Rajgopal Rudrarupu from ALMAC.
Yeah, thanks Roshan for introducing me. Hello everyone. Today I'm very excited to present the topic on prior knowledge and its benefits for the biopharmaceutical industry. However, this session predominantly focuses on CAF analytical methodology that was performed at Almac Sciences. Coming to the session outline, today's session we cover on the complexity of biologics in biopharma industry and the prior knowledge and how it can be implemented, and also the case study of NISTmAb at Almac Sciences and on CIF analytical methodology and the key takeaways. So before we on the complexity of biologics, biologics are generally large and complex medicines containing active substances made by biological process or derived from a biological sources. For example, if you consider monoclonal antibodies, which are used in the treatment of cancers or autoimmune disorders, are produced majorly in the inside of living cells. So in the recent biologics are also produced through cutting edge biotechnology processes, like where if you consider the recent vaccines which are produced by Pfizer and Moderna, where mRNA vaccines, which played a major role during this COVID-19 pandemic, are produced through synthetic procedures. But however, because of the mechanism of action, they're also considered as recognized as biologics. So broadly speaking, biologics includes all the vaccines, hormones, enzymes, and monoclonal antibodies, cell and gene therapy products, and also blood-derived products. So as biologics is very broader term, and for manufacturing and release testing of biologics is quite complex. So and due to we have due to challenging unit operations and analytical methodologies, but however leveraging prior knowledge helps to expedite CMC development of the molecule. So what actually the prior ICH guidelines tells about the prior knowledge. So if you look at the ICH-Q11, it includes that establishment that we can consider the established principles or technical literature or applied manufacturing experiences, and these aspects can be considered as prior knowledge and can be implemented in the manufacturing or in the during the process techniques in the industry level. And Q8 and Q9 and Q10 aspects, so it also tells us the experience obtained from the similar processes, like the internal knowledge or any industrial scientific knowledge or any technical publications. And also it also emphasizes that published information can also be used as a prior knowledge. So actually, where can we use this prior knowledge? In what areas can be used? It can be used in analytical area, where we can use for analytical method development and analytical validation. And it can also be used in process area, where we can go with process development and process validation. And it can also be used in strategic design, where the control strategy design or the development strategies can be established using prior knowledge. And also stability strategies can also be established. Apart from this, we can also use the prior knowledge during tech transfer within cross-site or within from R&D level to the QC level. And for our purpose, here we have published information from NISTmAb. This was used as a prior knowledge to establish CIF analytical methodology at Almac Sciences. So what exactly the NIST mab is? So NISTmAb is it's a reference material that was published by National Institute of Standards and Technology, and this NISTmAb can be used as a reference material and in various assets like we can use in method validation or method transfer or control charting, and also it can be used in analyst training for the biologic work. And with the very vast publication available on the NIST, so we have like tabulated the available methods on the NIST, like based on the, we have categorized all the methods on charge heterogeneity, size heterogeneity, and hydrophobic variance. And apart from all these methods, we have like considered the capillary isoelectrofocusing method for our case study. So actually this method was considered because this method can be used as an identity and as well as also for the charge variance establishment method. And apart from that, as this method also involves a complex sample preparation steps, so this method was considered as a case study at our laboratory. So uh, so this slides like shows up the method that is like published by the NISTmAb and NIST. And so actually the apparent PI of the NISTmAb is 9.18, and the main group percentage is 72.5, and acidic is near to 20%, and also the basic percentage is 7.5 percentage. And based on the available method parameters, we have like figured out the key differences which are like available in the existing SciX method, which is like latest published SciX method, and we have like brought down like key differences on the method and the difference in the methodology. So actually the NIST publication which is published, the data which is available, it's on 32 carat software, but at ALMAC we have an integrated Empower software with our CE instrument. So we have taken the latest publication of SciX, and from that the method parameters have been derived for the Empower. And we have considered the sample preparation part exactly same as NIST publication. So there was no change in the sample preparation part. And in this method, there were like few steps where urea and water rinse was performed prior to the injection, and whereas regarding with the latest publication, the sample loading solution and the water rinse was used. So we have used the same sample loading solution and water rinse as per the SciS publication. So these key differences were considered, and we have executed the method using Empower software and the CE instrument. So so by when we have executed, we have performed a method feasibility experiment, and we have got some inconsistency electropherograms. So as we have got like inconsistent electropherograms, immediately we had a look at the current profiles to check like what might have gone wrong. So when we had checked the current profiles, we have like seen there was a fluctuations observed in the current profiles. So so to understand the cause behind like the inconsistent profiles, we have break down few aspects in the troubleshooting. So the troubleshooting was done in step-by-step way. So we have like first taken the aspect as instrumentation. So the instrument will be the prior thing, like where it will be like it will be like adding to the changes in the current profile. So we have like sorted out like the voltage leakage and the performance are the key factors of the instruments. And in order to minimize the voltage leakage, or in order to nullify the voltage leakage, so we have cleaned all the opening levers and the electrodes of the instrument, and we have checked the performance of the instrument using test mix B solution, which is available from the SciX vendor. So when we perform this, like we got that like there's no impact on the profiles. So we have observed the same inconsistent profiles. So as the first step, where we have like figured out that there's no problem with the instrument, later on we evaluate the capillary, where for this methodology we'll be using a neutral capillary. So the capillary is like it's internally coated, and we were suspecting that there might be some damage to the internal coating. So in order to evaluate the capillary, we have taken a different lot of the capillary, and we have performed, but there was no impact on the results. Like we have got the inconsistent results there. So as instrument was fine and the capillary was fine, so next step, like we thought of, like we'll go with all the fresh reagents and the chemicals. So we have used all the fresh reagents and chemicals. But there was no impact on the results. So as we figured out the step by step, so later in our next thing, we have collaborated with the SciEx SciEx organization, and where the CE, where SciEx is a vendor for the CE. So we have collaborated with SciEx, and we have done some series of experiments, and we drawn an hypothesis that they because during our sample preparation, we'll be filtering the urea gel buffer using PVDF membrane. So we drawn an hypothesis that there might be some leachates that coming out of from this PVDF membrane, and that is causing that is generating some EOF in the capillary and causing a shift in the profiles. So later we figured out, we sorted out using either regenerated cellulose filters or to use no filtration, and we ensured that no visible crystals or no air bubbles are present, and we have executed the method feasibility experiment, and we got repeatable results. So this was done with the collaboration with the SciEx, and finally we have achieved consistent profiles of using the CAF methodology. Once the method is established, that we got an acceptable profile. So we did a comparative assessment with the profile that with what NIST has published. So here the topmost is a profile where the NIST is published, and the bottom is the ALMAC data that we have received at our lab. So if you observe, the acidic group has better resolution when compared to the NISTmAb profile. So furtherly, we figured out what might be the differences that we did, though the method parameters were considered from the SciEx publication, but still we see a better resolution at ALMAC. But once we checked out the data rate for NISTmAb was used as 2 hertz. The data rate what we used was 4 hertz. So to ensure that this might be the issue, so we have changed the data rate from 4 hertz to 2 hertz, and we repeated the feasibility experiment, and so that we got a profile which is comparable to the published profile by the NIST. So once the profile was comparable, we did a integration activity where the NIST publication did with the 32 carat, and ours was with the Empower integration. So the top 2 profiles, electropherograms, and to the left, which is NIST, which is done by 32 carat software, and to the right, there is Empower auto integration done at ALMAC. So the integration there was a difference in the integration when you do an auto integration with the Empower. So in order to mimic exactly with the 32 carat integration of the NISTmAb, we have done a manual integration on the Empower, and based on that, we gotten values of main region, acidic region, and the basic region. And if we compare the 4 hertz and 2 hertz, the 2 hertz manual integration figures were comparable with the 32 integration. 32 carat, which was performed by the NIST. So after once we were okay with the test method of the CIF methodology, we have performed all the parameters linearity and LOD, LOQ, and we have compared all the values side by side with the NISTmAb. So all that is successfully, it's indicating that we have successfully implemented the CIF methodology, which is more similar to the NISTmAb activity. And so during this process, while we were using the Empower, so we have faced multiple challenges when we were about to use the Empower. in case of 32-carat software while executing the experiments. So in the Empire, so this was a method parameters that were published by SciEx. In the row 4, if we observe on the column 2, so the event is for the sample injection where the capillary will be filled with the sample, and on the column 6, like the event which it takes the injection from the sample tray. So in this regard, the sample is like the the instrument takes the sample exactly from the vial specified. Like in the picture, it's like A1. So during this method, it says like we need to create a multiple methods to have an incremental injection, where with this method incremental injection is not possible. So for that reason, we need to have a multiple methods to go with the many like more than one sample. So for that, like we have changed the command from sample to sample list, where the sample list enables to have a incremental injection. So we have done that like when we started the execution of the experiment. So we got an error, like the sequence got aborted due to the tray error collision. So this was a bug that we identified during execution of the experiment. So later we figured out like few events that are present to fill the capillary out, or to take the injection of the sample into the capillary. So finally we have chosen a command, an event called like inject pressure, to take the sample inside the capillary. And we also changed the sample to the sample list, and and we have like slightly modified, as we have the cap of duration of injection time, like, and in the actual method it was 150 seconds, but when we were using inject pressure, we have only 99 seconds there. So for that case, we have increased the pressure from 15 psi to 25 psi, and we have decrease the time to 99 seconds. And with the modified method, we were able to execute the method, and we were able to implement the method with the prior knowledge available from the NISTmAb. And so these are the key takeaways, that we have utilized the NISTmAb prior knowledge, and we have successfully implemented the CAF method, analytical method. And we faced few challenges, but the prior knowledge helped us a little. It helped us, and we were able to implement the method. And we also did a transition from 32-carat to Empower software. And so this was the activity that we performed at Almac Sciences on using the prior knowledge of which is available from the NISTmAb. And we have like successfully established the method at Almac Sciences.
Great. Thanks.
Yeah.
Sorry about that.
Yeah, thank you.
Had to make sure that I was off mute. Okay. So
Okay.
the first question that we have is, what are typical issues observed during CE method transfers?
So generally, while we transfer a CE method, so there are like multiple issues that we face. Like first, like we can segregate it like instrument part, and then comes to the analyst part, and third we can go with the sample preparation part. So when we consider the instrumentation part, we need to at least like consider the environment where the instrument is placed. So we need to check out the temperature of the laboratory, and we need to check the humidity. And coming to the sample preparation part, we need to have a stepwise proper written SOP, where like if we miss like few steps out, so we might get a different profiles there out. And we need to have a proper training to the analyst in order to like have an ensure like proper method is being transferred to the lab.
Okay. And what do you think of strategies to implement prior knowledge in analytical labs?
Generally we don't have any strategies written by ICH, but the thing is that we can follow like 3R principles, like relevance, where we need to establish proper document, that like we need to see the similarity between whatever the project that we are going to take at our laboratory, and also we need to have a similarity comparison before we going up with the establishing a method or something kind like that. So we need to have a similarity documentation, and we need to also do a replicates, like we need to do a method performance assessment, and we need to also do a method knowledge matrix, use the control chart trending or statistical assessment. Like all these tools will help in uh, figuring out like whether we can use prior knowledge to establish the method or not in our analytical lab.
Okay, great. Well, it looks like we have time for just one more question. You can continue to type your questions in for Raj, and we will pass them along to him, and he can follow up with you directly. But the last question for the webcast is, what are the advantages of upgrading from 32-carat software to Empower?
The advantages what I see is that so Empower is like it is like support. It can support Citrix, so like we can do remote work from like using Citrix, and Empower has a centralized data storage where all the data can be stored at the center location, and we can also do a easy batch processing there because in 32 carat we can't do a batch processing, and we have a well established report templates for the in Empire, so reporting will be easier when compared to the other softwares.
Okay, well, thank you, Raj. And
Yeah.
thank you to our audience for joining us. The recorded version of this webcast will be made available for replay on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Yeah, thank you.